Okay, our next speaker is going to be Dr. Chia Lee. And quick introductions, Dr. Chia Lee is a research or equivalent to associate professor in biological chemistry at the Imperial College London. She was trained as a chemist at Nankai University, China, and obtained a PhD in biochemistry specialized in metabolomics at the Universities of London in 2009. Her research works and her research groups focus on the host diet microbial crosstalk and to explore the functions of the gut luminal metabolites in maintaining our health. She also received numerous awards and prestigious fellowships, including Imperial College Research Fellowship, Medical Research Council New Investigator Grant, European Research Council Starter Grant, Mueang Research Fellowships, and LC's Wilderson Fellowships Award, and many more. Dr. Lee is also a member of Royal Societies of Chemistries and the Biochemical Societies. Apart from all these research and awards, Dr. Chia Lee also plays a significant role in mentoring and training many of the graduate students here at the Faculty of Medicine, Khon University. And it's been a pleasure to welcome you back to Khon. So please give her a big hand of applause and welcome Dr. Chia Lee on the stage.
Good afternoon, everyone. Thank you very much for the invitation. It's really my pleasure to be here as always. Just waiting for my slides to show up. So today I'm going to talk about exploring the functions of the host microbial co-metabolites. As Jeremy Lane touched on this topic earlier this morning, so the host, the gut microbiota, and the diet have very complex interactions among the three. Just to re-emphasize that, for example, the diet we eat can deter or influence the metabolic activity of our gut microbiota, and they produce a set of microbial metabolites, which can in turn influence our phenome, or gut physiology and the pathophysiology. Through this interaction, a set of really complex molecules are generated. So here you can see this blue bubble here representing the gut lumen. You can see the dietary choline, which can be converted to methylamines by the gut microbiota. And for example, fiber can be fermented by the gut microbiota to produce short-chain fatty acids, which are believed to be beneficial for our gut health. There are also protein digestion. Can't see my pointer very well. Protein digestion, fat digestion, and some of the medium-chain fatty acids have been shown to suppress the growth of the gut bacteria, which could serve a new generation of antibiotics potentially. There's also bile acids metabolism. The gut microbiota can produce secondary metabolites from the primary bile acids. So all of these metabolites can be taken into the body to different locations, different organs, influence our health, disease. So to look at this complex set of molecules, we use metabolic phenotyping. Here you can see an example of proton NMR spectra here. All these peaks representing different metabolites that come in from endogenous metabolites, from our diet, or from the microbial metabolites. It also shows the information about host microbial co-metabolites. These are a set of metabolites that are generated by the collaboration between the gut bacterial genomes and the host genome. So I have applied this approach to look at surgical management of obesity. As you know, and also Jeremy mentioned, obesity or overweight or obesity is a major health problem in the world. And you can see on average, 40% of the population are either overweight or obese. In some countries, this percentage can reach 80%. So surgical treatment is the most effective treatment for morbidly obese. So that means the BMI is above 35. And you can see there are a few different types of bariatric surgery. The gastric banding is the reversible procedure. And simply just put a band on top of the stomach and you can tighten or loosen this button here. The second one is sleeve gastrectomy, where the 3/4 of the stomach is removed. This is an irreversible procedure. And the Roux-en-Y gastric bypass, or RYGB, is the most complex version of bariatric surgery. Is also a gold standard in the past 10 years also. And you can see here a small gastric pouch is created. The majority of the stomach and duodenum is bypassed, and jejunum is connected to this small gastric pouch, and the end of the duodenum connect to the lower part of the jejunum. So as a result, when you eat the food coming down this path, and the pancreatic digestive juice will come down from this limb and get in contact with the food much later on in the normal situation. So these procedures are very effective in terms of reducing body weight. It also can reduce various disease risks, for example, cardiovascular diseases, various cancers. It can also improve up to 80% of diabetes within weeks before the majority of significant weight loss happens. The surgery can increase the gut hormones, especially glucagon-like peptide 1 or GLP-1. For a very long time, people think the gut hormone changes can explain the surgical outcomes. So you can see here, it shows that 3 different time points pre-surgery, 6 weeks post-surgery and 12 months post-surgery. And regardless, patients are stratified in different groups, either normal glucose tolerance or impaired glucose tolerance or type 2 diabetes. Their gut hormones are all improved, or GLP-1 all increased post-surgery. So really test whether the gut hormones can explain all the surgical outcomes. We conducted a study to infuse a set of gut hormones to mimic the effect of RYGB into the patients without the surgical treatment. As you can see, we included 4 different groups of patients, saline as absolute control, and we have gut hormone infused, we call it GOP group. And we also included a caloric restriction that mimicking the amount of calorie patients have after bariatric surgery, and we also include RYGB group. So you can see here, this is the weight loss post the different interventions. You can see a GOP group has a weight reduction. However, the degree of the weight reduction is much lower compared with the caloric restriction and RYGB group. Then we went on to look at metabolic responses of these interventions using NMR and LCMS, followed by multivariate statistical modeling. Here I show you a PCA plot. So you can see here, RYGB patients' metabolic profiles all clustered together, and they all showed higher host-microbial co-metabolites post this intervention. And you can also see a cluster of patients here, which are coming from very low calorie diets. And the ketone bodies are one of the signatures coming up. So we know ketone bodies are features of fasting. But if you look at the GOP group in red dots and the control saline in blue, you can't really see much clustering patterns here. So that indicates the RYGB surgery has its unique metabolic features. And those features could be linked to the changes in the gut microbial function or composition. So subsequently, we wanted to know why those metabolic changes happen and whether they can contribute to weight loss after surgery. So we looked into one of the RYGB cohorts with much longer follow-up. follow-up. So you can see we have 2 to 6 months intermediate follow-up and 1 to 2 years long-term follow-up. And we did, again, NMR and LCMS analysis for the biofluids, as well as 16S RNA gene sequencing to look at microbacterial composition after the surgery. So you can see the patients all achieved sustained weight loss across the 3 time points. And if you look at their bacterial composition, so here we can look at the beta diversity of all the samples. You can see the pre-surgery, the battery's gone. The pre-surgery in black, the bacterial compositions are clearly separated from the post-surgical time points in green and orange over there. So these separations mainly caused by increased Bacteriaceae family in the gut microbiome. And I picked up 2 typical genus representing here. You can see Klebsiella and Escherichia shigella. They both increase significantly post-surgery compared with pre-surgery. Then the next question we asked is that whether these change the microbiome bacterial increase can contribute to weight loss after the surgery. So to test this, we took the microbiome from RYGB or sham operated rats and transplant them into a new set of animals which didn't have any surgical intervention. And then we followed up them for 16 days after fecal transplantation or FMT. And first thing first, we wanted to see whether there's a weight changes. So very disappointing that we didn't see any changes in their weights. You can see across the whole 16 days, there's no significant difference in body weight between the RYGB recipients and the sham microbiome recipients. If you look at the overall changes across the 16 days, you can also see there's no differences. In fact, the RYGB microbiome recipients have a slightly higher body weight. So that's quite contrary to what we hypothesized or expected. Then we thought, okay, how many we look into the microbiome? Did all the microbiome from the donor actually grow in the recipients? So you can see here, we plotted them at each time point. So we collected some samples at day 1, 3, 6, 9, and 16 days post FMT. And you can see a clear separation between the pre-transplantation and the post-transplantation. That indicates some of the microbes or bacteria can or actually colonize in the recipient's gut. As the days grow to the 16 days here, you can also see this start separation between the RYGB recipients and the sham recipients. We thought that's quite exciting. Perhaps some of the microbes have already colonized there. So we looked into details. So we found about 32% of the ASV, so amplicon sequencing variants, so representing the different bacterial group that colonized from the donor to, in the recipients of the RYGB microbiome. However, it's only 11% from the sham microbiome colonized in the recipients. Then we looked into exactly what species or a genus has been colonized in the gut. That's very disappointing to us. Again, that's Enterobacteriaceae, which was the interest of our research, didn't colonize in the gut. This suggests that this typical group of bacteria perhaps only prefers the environment that's created by the bariatric surgery, and it can't really be competitive to other bacteria in a normal gut. Therefore, we can't really see the colonization of this Enterobacteriaceae family, and we can't really answer the question whether this bacterial family can contribute to weight loss or not. And then we thought we also maybe look at the metabolic changes. Is there any changes? Despite the 32% of the ASV has been colonized in the recipients, we can't see any of the metabolic changes in the urine or feces. This again suggested that the bacterial function redundancies is very clear in this study. So in a way that, in other words, so different bacteria could come to do exactly the same metabolic job in our gut. So this message made me think, okay, well, instead of trying to manipulate the gut microbiome composition, why don't we just change the metabolites concentration in the gut and see what it does to the gut physiology? So before we do that, we first look into the metabolic changes in those biofluids coming from bariatric patients. So again, you can see a clear separation between the pre-surgery and the post-surgical time points. And if you look at those loading plots representing the key metabolites that's contributing to this difference, you can see phenylacetylglutamine, PAG, indoxysulfate, parathiazole sulfate, all of these metabolites are host microbial co-metabolites, together with trimethylamine oxide or TMAO. I'll come back to this molecule in a bit. Then we also look at the fecal metabolites, and you can see here, again, similar to the urinary profile, you can see the difference between different time points. And if you look at the metabolites that's contributing to those differences, the two metabolites here, phenylacetate and tyramine, are elevated significantly after the surgery. Then we wonder whether this metabolites increase is due to change the microbiota. Then we took the fecal samples from a healthy donor and RYGB donor. We batch cultured them in the lab with the complex media containing tyrosine, which is a precursor for tyramine. And after about a day or two, from the healthy donor, we can see tyrosine peaks here, and there's no other metabolites produced. But if you look at the RYGB donor, you can see tyrosine is here, intensity is reduced. Instead of, you can see also tyramine peaks showing up. That means that the tyrosine can be converted to tyramine through by the tyrosine decarboxylase present in those bacteria. That's probably most abundant in the RYGB donor. But if you look at this side of the spectrum, butyrate, as I mentioned earlier on, is believed to be beneficial to the gut epithelial. And you can see in healthy donor, butyrate's production is much higher compared with RYGB donor. So all these metabolites patterns make us think this is not a friendly environment. It could be pointing towards an unhealthy gut environment after bariatric surgery. Then we also looked into the literature. We found this paper published now 10 years back. It's a very large study looking at bariatric surgery patients' risk, colorectal cancer risk. They actually reported that patients with bariatric surgery had much higher colorectal cancer risk compared with the obese individual without surgery. Even though this sort of finding is still controversial up to now, there's still debate in the field. But what we're really interested in is whether those molecules we see from the fecal water could contribute to this disease risk. So we first focused on tyramine, which was significantly changed in the fecal water metabolome. And you can see tyramine not only can be produced by the gut bacteria, but also produced by the bacteria we use to ferment food. As you can see, tyramine is highly present in salted cod fish that's used in Portuguese cuisine, and also fish sauce, which often used in the Thai cuisine as a fermented cheese and ham and so on. So we first treat HCT116 cell, human colonic epithelial cells with tyramine at various concentrations. And we use the micronuclear assay to evaluate the genotoxicity property of tyramine. And you can see here, you can see a little nuclear is derived from the major one as the cell divided. So we count those micronuclear assay. You can see at around 1.2 millimole treatment, there is a significant genotoxic effect induced by tyramine. Then we also use a different marker to evaluate this observation, which is gamma H2AX, which is marker for DNA damage and repair. And again, can see at this concentration, 1.2 millimole, it has a significant increase compared with the control group. And another marker, which is a known risk marker for colon cancer, is the cell proliferation. The higher rate the cell proliferates, the higher risk you might have. So that is the marker of Ki-67. And we look at this, you see at the treatment of 1.2 millimole, you can also see a significant higher cell proliferation compared with the control. So these results make us think maybe tyramine can promote colorectal cancer tumor progression. Then we next test this in a mouse model, genetically modified mouse model, APC min mice. It's not the best model for mimicking human colorectal cancer, but it was the best genetic model at the time when we did the experiment. So we fed the mice, both wild type and APC min mice, with tyramine every day for 49 days. And we called the mice, counted the tumor. You can see those some examples of a tumor we spotted in their intestine. And we count the tumor in each part of the intestine. And you can see it's not surprising that APC min mice have much higher tumor load compared with the wild type. However, I just want to draw your attention that even though there's not reaching statistical significance, but you can see there's clearly higher tumor instance in the tyramine-treated APC min mice compared with the water-treated APC min mice. And here you can also see even in the wild type treated with tyramine, there are also 2 mice present to the tumor compared with the water-treated wild type mice. And we also looked into the immune landscape in the gut tissue because inflammation is one of the drivers for cancer tumor formation. So here we screened quite extensive different set of immune cells. And what we found was a suppression of the natural killer cells after the tyramine treatment. And if you look at this NKG2D, which is representing activated version of NK cells, as clearly reduced after tyramine treatment. This suggests that because NK cell is the type of cells that's showing the anti-cancer property, and tyramine can reduce the anti-cancer property, which promotes the cancer development. And on the other hand, so you can see this one, the PMN and DSC has got a long name here, polymorphonuclear myeloids derived suppressors. So this type of cells also, they suppress the anti-cancer properties. So that means when they are elevated, they can promote cancer development. So you can see those observations together showing that tyramine can create a tumorigenic immune responses in the APC min mice. And similarly, we saw a very consistent evidence from neutrophil as well. So we did RNA-seq analysis of the ileum tissue, and one of the protein here, the expression level of MRA, that's from neutrophilic granule protein, and you can see that's a highly present, expressed in the tyramine-treated mice. And we also confirmed these results using immunohistochemistry method. You can see a clear significant increase in the neutrophils in the ileum samples. So what really interesting in this study, in addition to increase the tumor load or increase the inflammatory responses in the tyramine-treated APC min mice, is actually in the wild type animals. We found that the cell proliferation also have a tendency of increase after tyramine treatment. And if you look at the genotoxicity or DNA damage marker gamma H2AX again, that's also showed a higher level of DNA damage and repair after tyramine treatment in wild type animal. So this evidence suggests that tyramine could increase the colorectal cancer risk in a wild type animal. So now move on to the next molecule, TMAO, trimethylamine oxide. As I mentioned earlier on, it elevated after bariatric surgery. So Jeremy Nelaine published a very comprehensive review on this molecule last year. And in my study, we mostly focus on the association between TMAO and colorectal cancer. So TMAO that can be derived from dietary choline or carnitine, which presents high levels in red meat and egg yolk. And these molecules can be converted to trimethylamine by the gut bacteria. And trimethylamine can be taken into the liver, get oxidized by this enzyme FMO3 to TMAO. And TMAO can also, very small fraction of that can come back to the intestine and get converted back to TMA by the gut bacteria. This is so-called retroconversion. So what we're really interested in is whether TMAO that's elevated after bariatric surgery could also contribute to the colorectal cancer risk. There are some studies showing there might be association between them. Oh yeah, so just this to demonstrate TMAO is not only increased in the urine samples, but also in the plasma samples post-bariatric surgery. And it has been shown that this increase is nothing to do with the dietary modification post-surgery. So we think this increase could be linked to the bacterial changes. So we isolated the bacteria that's elevated in bariatric patients, for example, Klebsiella oxytoca or Enterobacter cloacia. So we culture those two bugs using a simple carbon, single carbon media, which contain choline only. And you can see here, this is a control sample. You can see a choline peak here. And when you have samples that's having those bugs present, they convert choline into trimethylamine here. So it's clear that in the bariatric surgery scenario, the increased TMAO is very likely to be linked to the increase of those Enterobacter SAI bacterial family. And this paper also showed that the key enzyme that's responsible for choline to TMA conversion, a choline trimethylamine lyase gene, was abundant much higher in the CRC colorectal cancer patients across, I believe, 6 different cohorts across different countries. So this information collectively led us to hypothesize the increased TMAO in bariatric surgery, mainly to increase colorectal cancer. And this point, we use a different genetically altered animal. So we spent 3 years here to create this model, which is better mimicking human sporadic cancer, colon cancer, because it's mainly having the tumors in the large intestine, and also the number of tumors is much less compared with the APC in mice I mentioned early on. So we give those mice either controlled diet or TMAO supplemented diet for 6 or 8 weeks in male or female respectively. And then we count them at the end of the treatment. We count the tumor, we look at the flow, we look at the immune cell functions in the gut using flow cytometry. So again, to our surprise, the findings is opposite to our hypothesis. So TMAO supplements actually protect against the tumor growth. And you can see here the percentage of animals that's present, that have a tumor presence is much higher in the control, 94.4%. In contrast, the TMAO-fed animals have only 53%. And this protection role, you can also see very clear in male mice compared with the female. There's slight reduction, but not as dramatic as in male. If it counts all the tumor volume within each animal, you can see there's a clear significant reduction in male mice fed with the TMAO compared with the control. And there's a slight reduction in female, but not statistically significant. Then we looked into the immune profiles, and you can see here TNF alpha levels draw our attention. You can see in 3 different subsets of immune cells, CD8 positive, CD4 positive, and natural killer cells. The TNF alpha levels are consistently lowered in the TMAO group compared with the control. And only happens in the male. If you look at female, there's no such a reduction trend. So what we think at the moment is that the TMAO could provide the protection role possibly through the suppression of TNF-alpha expression in the immune cells in the gut. So now we move, another PhD student in the group is also really interested in TNF-alpha function and how that associated with the colon cancer. So what she did was to treat TNF-alpha at the physiological and the pathophysiological concentration to the HCT116 cells, and she found a significantly higher genotoxicity induced by TNF-alpha. And then she went on to see which signaling pathways involved in this TNF-alpha mediated genotoxicity. So first she looked the nuclear factor kappa B signaling. So she used inhibitors to inhibit this pathway, and you can see the genotoxicity is not reduced or not changed compared with the TNF-alpha treated group. In contrast, if you look at this one, the gene K pathway, when you have an inhibitor to inhibit the gene K signaling pathway, the genotoxicity is significantly reduced. So the results are showing that the TNF-alpha mediated the genotoxicity is through gene K signaling rather than an alpha kappa B pathway in this particular HCT116 human epithelial colorectal cancer cell line. So as you can see, inflammation is really a key factor in colon cancer. And another student in the group who's a clinical research fellow, she's very interested in inflammatory bowel disease, especially ulcerative colitis. So you can see this disease mostly affects colon and rectum. And there's different types of UC here. And the prevalence of UC is also very high, especially in developed country like in the US, Australia, and also UK. The reason of people developing UC is unclear, but generally people believe that's a sort of interaction between environmental factors and genetic predisposition, plus impaired immunity and also microbial disorders. What she really interested in was one of the tryptophan metabolites. So it's called the indole-3 aldehyde, or IALD. So you can see there's a complex tryptophan metabolites, and these metabolites can be produced by, I believe, lactobacilli bacteria from tryptophan. And this is also present in some of the very healthy vegetables like broccoli or white cabbage and Brussels sprouts, which are quite famous in the UK for Christmas. So what we really look into is whether this compound IOLD can mediate the inflammatory responses through AHR signaling pathway. So AHR is a aryl hydrocarbon receptor. So this AHR signaling pathway actually mediated in many different cellular functions, as well as the inflammation. So she has done quite a lot of cell and animal work. Today I'll just show you a fraction of her animal results here. So she included 5 different groups. So the olive oil here is actually acted as a vehicle control, and then she gave another group the IOLD only, and the other 3 groups show induced colitis using this chemical DSS. Before inducing the colitis, she primed those animals with either a vehicle or IOLD, or IOLD with a AHR inhibitor called the SAGE-22 for short here. So after about a week after colitis, she culled the animals. So you can see here is the body weight changes across the whole experiment. The red lines are the animals with the DSS with the colitis without any other treatment. You can see the body weight drop very significantly. When the animals were primed with IOLD, and their body weights remain relatively okay, not as high as the non-colitis animal, but it is significantly improved compared with the colitis animals. But when the animals are treated with IOLD plus the AHR inhibitor, the phenotype of body weight increases disappeared. So similarly, you can see the gut length, this colon length, again, with IOD treatment, this bar here, you can see there's a slight improvement, but significant. And when you have a co-treatment of IOD plus inhibitor, this phenotype also disappeared. And what's really interesting is 3 pro-inflammatory cytokines that's involved in UC disease. These are IL-6, IL-1 beta, as well as TNF alpha. So she measured those 3 cytokines in the gut, in the colon, and you can see they all reduced after the ILD treatment. You can see here compared with the DSS treated animals. But if you look at the inhibitor group here, in the IL-6, you can see when you apply the inhibitor, the IL-6 levels bounced back. That means the IOD mediated the IL-6 reduction is through the AHR signaling pathway. In contrast, if you look at these 2 groups here, when you apply the inhibitor, there's no change between the IOD alone group and the inhibitor group plus IOD. That suggests that the IOD does reduce these 2 pro-inflammatory cytokines. However, it's not mediated by AHR signaling pathway. So, I think I was almost done here. So just to summarize the take-home message of my talk, as you can see, our IGB surgery induced really significant changes in the gut microbial composition, especially increased Enterobacteriaceae. However, whether this bacteria can reduce weight loss remain a question to be explored in the future. And the co-metabolites, host microbial co-metabolites, are significantly increased post-surgery. Those are including TMAO, paracrystal sulfate, phenylacetide glycine, and so on. And also examples I show here are the timing, which induced the DNA instability and cell proliferation, which are 2 known risk markers for colon cancer. And TMAO, which our findings are quite opposite to what's been reported so far in the literature. We found TMAO is actually showing a protective role against the tumor development, especially in male compared with female mice. And lastly, this tryptophan metabolites in those 3 aldehydes can reduce the pro-inflammatory cytokines, and only the IL-6, which is through HR signaling in this DSS-induced colitis model. So I think I just wanted to finish with these slides. I showed at the very beginning of my talk. In all my experiments, we treated the mice with a single metabolite. I know that's not really the reality. The reality is that we exposed to all these molecules at the same, more or less the same time after a meal. So I think really the research going forward is to look at how those molecules actually interact with each other, how they interact together with the host to influence the gut physiology and the health. So finally, I'd like to thank my collaborator, especially Nadia, who's an immunologist, to help a lot with these studies, and Professor Nigel Goodrum, Elaine, and also Tricia Tan, who conducted the gut hormone study, and also my previous group members and the current ones who contribute to the studies I'll show today, as well as some funding bodies. So most importantly, thank you very much for your attention today.
Thank you so much, Dr. Shirley. Please give a big hand. So that shows us a really interesting stories of how the little microbes can have such a profound effect on our health and disease and well-being. And it's not just the microbes alone, or it's not just a particular part, but you also look at the metabolomics, the RNA sequencing, the gene expressions, and even immune system as well. So with that, was there any questions from the audience? Well, thank you so much for showing us all this information. It's beautiful to see how every story connects together. But if there's no question from the audience, just one thing, when we talk about all these effects and is linked to the previous talk from the Professor Chatchai. He was talking about his big data and the directions of this omic research, the need for bioinformaticians. So was there any challenge that you yourself or say the students that you train face when they work with this huge amount of data, various omics and the integrations, and how did you or your research team overcome these challenges of working with big data?
Couldn't hear the last bit,
right?
Yeah.
Yeah, let's try again. So you told us about these metabolomics and RNA sequencings and also the immune systems and all like metabolic pathway as well. It's a huge integrations and it's a big data. So I think many people would find that aspect very challenging. So my question would be, did you face this kind of feeling of like the challenge during your research, or your students, how do they feel about it and how did you overcome this challenge?
Yeah, it's definitely been a very challenging road. I mean, I'm a chemist, so really most of those studies are not really in my expertise. So it's really big learning curve, but I would just say that it's really important to have expertise, diverse expertise in the group. So for example, my postdoc, well, she left the group recently, Maria Kallaprapruek. So she's an immunologist. So really grateful to have her in my group for 5 years. So I think quite a lot of this work wouldn't be done without her. So I think, yes, like you said, it's been challenging, but I think when people work together and this sort of immersion, really involve people. I know it's challenging during COVID because everyone is working from home, but I think the concept of working together really interactively, it really benefits the research progress.
Yeah. Okay, thank you so much. Thank you so much. And please give us a big round of applause for Dr. Chia Lee. So big data, big research teams, and